Hello everyone. In this video we will discuss the topic clusters and tandem repeats, microsatellite repeat sequence, transposons, or we can say jumping genes. Transposons, the class of the genetic elements that can jump to the different locations in a genomes. Although these elements are frequently called jumping genes, they are always maintained in an integrated site in the genomes. In addition, most transposons eventually becomes inactive and no longer move. The transposons were first discovered in the corn maize during the 1940 and 1950s between by the American scientist Barbara McClintock, whose work won her the Nobel Prize in the field of the physiology or magicians in 1983. This is the picture of the Barbara McClintock. Now the characteristics of the transposable elements. So some salient features of transposable elements are as following. These are the DNA sequences that code for the enzymes which result in self-duplications and insertions into a new DNA site. The transposons are involved in the transpositions events which include both recombinations, replications, which usually generates the two copies of the original transposable elements. One of the copies remains at the parent site, whereas the other one reaches the target sites on the host chromosomes. The integrity of the target genes of these elements is invariably disrupted by the presence of those elements because transposons carry the genes for the initiations of the RNA synthesis. Some previously dormant genes might be activated. A transposable element does not have a site for the origin of the replications. As a result, it cannot replicate without the host chromosomes as occur in the case of the plasmids or in the phases. There is a no homology between the transposons and its target site for the insertions, and these elements can insert at almost any positions in the host chromosomes or in the plasmids. Some transposons might seem likely to enter at some specific positions, and these sites are generally called as the hotspots, and they barely insert at the base specific target site. Now we will discuss about the classifications. So since McClintock's discovery of the transposon, three basic types of the transposons have been identified. These types are the retrotransposons and also called the class 1 transposon. Second is the class 2 transposon or known as the DNA transposons. And third one is the miniature inverted repeat transposable elements and represented as the MITS or class 3rd transposon. Now the retroposons. The class 1 transposons are copied in two stages. Here the meaning of the TE is the transposable elements. First, they are the transcribed from DNA to RNA, which represented in the picture, and the RNA produced is then reverse transcribed to the DNA. This copied DNA is then inserted back into the genome at a new positions. The reverse transcription step is catalyzed by a reverse transcriptase enzyme, which is often encoded by the transposable elements itself. The characteristics of transposons are the similar to the retrovirus, such as the HIV, and the retrotransposons are commonly grouped into three main order. These retrotransposons with long terminal repeats and represented as the LTRs, which encode the reverse transcriptase similar to the retrovirus, And the retroposons, and known as the long interspersed nuclear elements, and represented as the line or LINE or L1S, which encode the reverse transcriptase but lack the LTRs and are transcribed by the RNA polymerase II. The short interspersed nuclear element, that is represented as the signs, do not encode the reverse transcriptase and are transcribed by the RNA polymerase III. The retroviruses can also be considered as the TE elements, and for example, after the conversion of the retroviral RNA into the DNA inside a host cell, the newly produced retroviral DNA is integrated into the genome of the host cells. These integrated DNAs are termed as the provirus. The provirus, it is a specialized form of the eukaryotic retrotransposons which can produce the RNA intermediate that may leave the host cells and infect the other cells. The transposition cycle of the retroviruses has the similarity to that of the prokaryotic Ts and suggesting a distinct relationships between the two type of the genomes or the organism. Now we will discuss about the second class, that is the DNA transposon. The cut and paste transposition mechanism of the class 2 TE transposable elements does not involve an RNA intermediate. The transpositions are catalyzed by the several transposase enzymes. As you can see in the diagram, some transposons non-specifically bind to any target site in the DNA, whereas other binds to the specific target sequences. In the diagram, it is clearly, you can see after the cleavage, both the fragments are joined together and form a mature strands. The transposons or transposase makes a staggered cut at the target sites and producing the sticky ends cuts out the DNA transposon and ligate to each other into the target sites. A DNA polymerase fills in the resulting gaps from the sticky ends and the DNA ligase closes the sugar phosphate backbone. This results in the target site duplications and the insertion sites of the DNA. The transposons may be identified by the short direct repeat and a staggered cut in the target DNA filled by the DNA polymerase, and it is followed by the inverted repeat sequences. Cut and the paste transposable elements may be duplicated if their transpositions takes place during the S phase of the cell cycle. S phase means the synthesis phase of the cell cycle, when a donor site has already been replicated but a target site has not yet been replicated. Such duplications at the target sites can result in gene duplications, which plays an important role in the genomic evolutions. Now, the autonomous and non-autonomous transpositions. The transpositions can be classified as either the autonomous self or non-autonomous in both class 1 and class 2 transposable elements. The autonomous transposable elements can move by themselves, whereas the non-autonomous transposable elements require the presence of the another transposable elements for movement. This is often because dependent the transposable element and lack the transposase enzyme or the reverse transcriptase enzyme for the class 1 and the transposase for the class 2. The activator element represented as the AC is an example of the autonomous transposable elements, and the dissociation element such as the DS is an example of a non-autonomous without AC, and DS is not able to transpose. Now the transposable elements are of the following two types. These are the IS, means insertion sequences, or also known as the simple transposons, and the second one is the transposons, represented as the TN or the complex transposon. Now the simple transposon or the insertion sequences. So as you see in the diagram, in the middle part is the genes for the transcriptions or the transpositions, and the terminal ends of this segments is a inverted repeats sequences. The whole structure is the simple transposons. The insertion sequence or the simple transposons are shorter sequences which is about 800 to 1500 base pair long, but do not code for the proteins. These sequences carry the genetic informations which are the necessary for their transpositions, that is, the gene for the enzyme transposes. Insertion sequences have been identified in the bacteriophage, in the F factor plasmids, and many bacteria. Now the transposons or complex transposon. The complex transposons are the several thousand base pair longs and have the genes that code for one or more type of the protein. As you can see in the diagram, this is a very complex structures and have the many genes for the synthesis of the proteins, and which might include the resistance factor in the bacteria which acting against the antibiotics. A distinguishing characters of the transposon is the presence of the identical inverted terminal repeat that is represented as the IR. These are the sequences which is ranging from about 8 to 38 base pairs. Now we will talk about the examples of some transposable elements. So the examples are the TN3, transposable element 3, which is transposon of the E. coli. Transposon 3 has 4957 base pair and contains the three genes such as the TNPA and TNPR. TNPA means transposon A genes codes for the transposes having the 1015 amino acids and required for the transpositions. Transposons are codes for a repressor containing the 185 amino acids which regulates the transposes activity. BLA codes for a beta-lactamase enzyme which confers the resistance to the active antibiotic ampicillin. Now another example is the bacteriophage mu, and mu is meaning is the mutator. It is a temperate bacteriophage which having the usual phase properties and could be regarded as a large transposons. It inserts itself into the coli chromosomes at random locations and resulting in the. Now the applications of transposable elements. The transposable elements can be used as a genetic tool for the analysis of the gene expressions and the protein functioning. These are used in genetic engineering to insert or remove the specific genetic sequences and also to cause the frame shift mutations. The transposons, one mariner class of the TE Sleeping Beauty transposon system, is being studied for use in human gene therapy.